Hi. We are at the final process. And this is where we take what's known as the electron transport chain, which helps to utilize potential energy from electrons to produce a hydrogen or proton gradient across the inner membrane of the mitochondria. And that is going to help to activate ATP synthase to make ATP. Now, the process of making ATP through ATP synthase is called oxidative phosphorylation. And so the electron transport chain is very much coupled to oxidative phosphorylation. We do all of this, though, by taking the electrons from NADH and FADH2 and shuffling them into a series of redox reactions using the electron transport chain. So let's take a look at this so that we can understand this a little bit better. So as we take this, and you can see here is the outer membrane for the mitochondria. Here is the cytosol. Here, this is a cristae that's here, and here's the inner membrane, right? And so you're going to have these electron donors that donate electrons on these initial enzymes here. And then those electrons are going to move through here, giving those proteins or enzymes the energy they need to perform the functions that they are doing. And so by the time they get to the end of this process or this chain, they have little energy left. And so what happens is they get to the end of the road, basically, and so they need to go somewhere. And so what happens is that oxygen, and that's why we breathe oxygen, comes here, accepts the electrons, and combines with the hydrogen to make water. And so this is where some of our water comes from. We drink it, but we also have metabolic water we make. So let me blow this particular thing up so I can explain exactly what's happening here. So this first part right here is the electron transport chain. And they have big names for this. For instance, this is known as, if you look, complex 1. It's also known as NADH dehydrogenase. They're all basically enzymes, all of these. This right here, complex 1, complex 2, 3, 4, and this is ATP synthase. Guess what ATP synthase does? And so how we enter electrons or energy into this is either through what's known as this complex 1. It takes NADH, and remember Leo says GR, so we notice it's taking the hydrogen away and it's making NAD+. And so here you're removing electrons from here. So we are oxidizing that, and it's shuffling through. Now, this will move through on a little molecule that's like a shuffler that goes through here. You know, I do not like this picture. That's so much better. So let's look at how this works, essentially. So you can see here, this is a cytosol or cytoplasm. Here is the mitochondrial matrix. So as you can see here, this is the electron transport chain here, and then this is going to be basically oxidative phosphorylation happening here. And so here is the cytoplasm or cytosol here. Here is the outer membrane of the mitochondria, the inner membrane of the mitochondria, and the inner membrane space. And you can see this picture, they've basically cut it out and put it here. So essentially, we've had the Krebs cycle in this mitochondrial matrix, and we've noticed that it's made both NADH and FADH2. So those molecules shuttle over here, and you can see that the NADH is going to come in here. This enzyme that's right here, that's what all of these are too, to let you know they're all enzymes. They're going to come and they're going to oxidize NADH, which means they're going to steal the electrons from it and the energy that goes with it. And in the process, the electron is going to move through here, and the energy is going to be used to open this up and move a hydrogen across the membrane into the intermembrane space. Now, as you can see, this basically right here, they have numbers or Roman numerals on them. This is 1, this is 2, this is 3, and this is 4. They're called complex 1, 2, 3, and 4. But they also have other names from them. And you can see for the most part, they have ACE on the end of them, right? This one's complex 3 is a little different, but complex 4, they all have ACE. And they basically tell you this complex 1 is also known as NADH dehydrogenase. So it has a couple different names for it. Anyway, they tell you what they do. And so what happens is you have the electrons that go into here, and that pushes the hydrogens through here. Now, at the end, once the electrons go through this enzyme, they're like, I need somewhere to go. So there's a little molecule called coenzyme Q here, vitamin K. And basically what happens here is that it's like a little raft that goes back and forth between 1, 2, and 3. What I mean by that is once the electrons need to jump off of this guy because they're done, it'll jump on the coenzyme Q and move them over to 3. The same thing. So we have, this is almost like this is the front door into the mitochondria electron transport chain. It also has a back door. That's complex 2, succinate dehydrogenase. What that is making, and you'll notice this is the only enzyme that you see that is located both in the Krebs cycle and also in the electron transport. It's kind of cool. So FADH2 will come in here. We will oxidize it to FAD. And so we'll steal the electrons from that, and then the electrons will move onto the raft known as coenzyme Q and go over to 3. If you know, this one doesn't span the full range of the membrane, so that's why you don't see any hydrogens going across there. Now, once the electrons come over to 3, you'll have them both from complex 1 and 2, right? That will go into 3. At this point, it'll move here, and the electrons, essentially the energy will be used from them again to help push hydrogens across the membrane again. And then essentially it'll get to complex 4, which is the last one in the electron transport chain. You'll notice with this that, again, to get it from 3 to 4, you need cytochrome C. And all of these you can take as supplements too to help. And this is how they help provide more of these molecules here for this to happen even faster and more efficient. So those electrons will move on to complex 4, and they'll move through complex 4. As they do, you'll notice they will push hydrogens through as well. Not as much, though, here, because we are losing some energy. And at the end, what will happen is oxygen will come and accept these because they're done riding the roller coaster, basically. So they need to go off. So this is kind of like if this is the top of the energy period, it would be like dropping someone off at the top of a mountain, and energy wise, they run down the mountain, and at the end of the mountain, there's a ferry because there's only water there. They can't go anywhere else. So they have to jump on a ferry to go, to continue, moving and doing their job. So the ferry is like oxygen. And then that, along with the hydrogens that are around electrons, are going to make water. Now, how does this process right here, because I didn't say anything about making ATP, right? The only thing we've done with the electron transport is use the potential energy that was in these electron carriers, NADH and FADH with the electrons, to move hydrogens across. But what you'll notice is we have developed a hydrogen gradient here. What do I mean by that? We have shoved a bunch of hydrogens over into this area, and they don't have anywhere to go. So basically, they're going to keep pushing and moving over there until their location, being so high on one side than the other, is going to give another enzyme the energy to make ATP. And so what essentially that is you have ATP synthase right here. And it's going to open up and use the electrons, the hydrogens on this side, and it's going to give it its energy to activate this particular enzyme right here. And that's going to take ADP and convert it to ATP. So this is how electron transport here is tied to oxidative phosphorylation, or the process of making ATP through ATP synthase. Phosphorylation is adding phosphates. So that's why that process is called that. So this, as you can see, this intermembrane is very, very important, this intermembrane space, for allowing this process to happen. And so we have our starting molecules of NADH, FADH2, oxygen, and ADP, an inorganic phosphate, right? Nothing directly big molecules from the glucose anymore, only these little molecules. And then we're going to end with NAD+, FAD, water, and ATP. And this process makes, it's a range between 32 to 34 ATP per glucose molecule.